Hello students, today our topic of discussion in our botany subject will be the replication process. This is one of the important topic in our chapter molecular basis of inheritance. Overall, actually the chapter is very important, molecular basis of inheritance. Let's see what is the replication. Replication is the formation of DNA from the DNA. DNA forming DNA, that you call it as replication. Now, what are the other things given over there? DNA forming RNA, that you call it as transcription. RNA forming protein, that you call it as translation. This thing in your book is given in this way under the central dogma in molecular biology. DNA giving DNA, I again repeat, replication. DNA to mRNA will be transcription. mRNA to protein you call it as translation process. Fine. Now we have to focus among all these on this point, that is the replication process. Rest all the things have been given in your book stepwise sequences, and we will take in our today lecture only the replication. Then in our next session we will cover about the transcription and so on, right? Now let's talk about the replication process in detail. Right now this diagram is showing you one of the concept. What is that? That replication is a semi-conservative process. What do you mean by semi-conservative process? The word semi means half. Now what thing halfly? They are maintaining from parent to the progeny. That is one of the strand of its DNA, not both the strand. Can you see in this diagram? The red color strand is representing the parental strand, and blue color, which is formed after replication, will be the daughter strand, or you call them as replicas. So red strand will go in one progeny and second will go in another progeny, means it will separate. But one progeny will have one strand, but not both the strand of the parent, right? DNA will be double stranded, but progeny will have the one strand formed parental and one is a newly formed. That will happen in this progeny. So that's why we call it a semi-conservative process. And who had proposed this thing? That is by Watson and Crick. Watson and Crick proposed that thing, that replication must be a semi-conservative process, but they never prove it by any of the experiment. This thing experimentally was proved by the scientists Meselson and Stahl. Let's talk about little bit about this experiment, then we can proceed with machinery and the enzyme of the replication, which are used in the replication. Now in this experiment, you must remember that they are involving the organism, that is Escherichia coli, which is a bacteria, which is a prokaryote. It is having double stranded DNA, right? And what they did, they cultured this E. coli into the N15 medium, which is ammonium chloride. And N15 is not a radioisotope. This is an heavier isotope of the nitrogen. This is very important question. N15 is not the radioisotope. This is a heavier isotope of the nitrogen. And in that medium, they culture this E. coli for many generations. Once they get both the strands N15, N15, they select that as a parent for this experiment. Like, let me to show you in detail little bit. Like, suppose if I am taking this as a one E. coli, it is having the DNA inside it. Now, instead of showing the full E. coli, I am showing its DNA simply like this, which is double stranded. Initially, it is N14 strands. In natural form, it will be N14 and N14. But if you start culturing into N15 medium, then what will happen? When this will replicate, when this cell will divide, obviously it will divide. Before that, the DNA will replicate. So can I say this will separate and new replica is going to be formed, will be N15 and N15, because in the medium where you are replicating, that is the new replica, the nitrogen they will receive. Can I say it is going to be N14 and N15, N14 and N15. You can call it as hybrid. Now again put that into N15 only. Now can I say their strands are going to separate, right? Even the yellow one, they are going to separate, right? But once they separate, they should be double stranded. The new replica form will be N15 and N15. Right? Can I say in these cells which are having this type of DNA, that DNA is what thing? Hybrid again. But this will be the pure for the heavier DNA molecule. So we have to select. We have to select this as a parent now, because they're both the strands have become N15 and N15. Now we can say, we can start with this experiment, being both the strands have become N15 and N15. Now once this experiment was started, now they kept this into N14 medium. Once they kept in N14, can I say new replica will be forming N14 now? In which medium you are culturing, that will be the constituent of the replica formation, right? So this is a first generation which is having two molecules of DNA, or you can say one cell divided into the two cell, and this will take 20 minutes. 20 minutes for complete division of cell, but 18 minutes for replication. Rest 2 minutes is for the cytokinesis, means final division of the cell here. Now this will again undergo division next 20 minutes, means from here to here total 40, but here to here will be next 20 minutes. And now we are having the 4 types of, or I must say 4 DNA molecules in the 4 bacteria. This is a second generation. Now in third generation, how many molecules will come? Do remember, you have to apply the formula 2 power n, where n is for the number of the generations. Now right now I am talking about the third generation, 2 power 3. It will be having 8 molecules of DNA. And one DNA molecule is having 2 strands. So 8 will have 16 strands. I repeat my question. In third generation, how many DNA molecules will be there? 8 DNA molecules. Now keep in your mind, in every generation as a formula, 2 will be hybrid. Hybrid word is always for molecule here, not for a strand, right? So hybrid means it will be having N14 and N15 strands. In every generation, it will be 2 molecules. Like here in first generation, 2 molecules. Here even it will be 2. And whatever is left now, that will be having they are the lighter strands. These are the lighter strands, right? These are the lighter strands. Being we are culturing this into N14 medium. These are not heavier. N14 is a normal one, right? So how we will come to know that this is going to be a normal or lighter strand, or which one is heavier, which one will be hybrid? For that thing, centrifugation is done. When you are doing the centrifugation, if it is coming as a sediment complete, that means it is the heavier one. It is a heavier molecule of DNA which is coming at the sediment. And if you are centrifuging that, if it is coming in the center, that means it is a hybrid. If it is coming at the top, that means it is a lighter one, right? So again, I repeat this one at the last, we are getting the lighter strands, right? Being we are culturing in N14 medium, and every generation must have 2 hybrids, and the rest over what is left will be the lighter DNA molecules, being we are culturing in N14 medium over there. Now, how we can come to know that this experiment is discussing about semi-conservative process? Because in no generation, you will get something at the base, means as a sediment. Why? Because this will be shown as both the strand and N15 only in parent. And after that, they will separate. One progeny will have the one strand, second will have the another. One progeny cannot have both the N15 in any of the generation. That's why you will not get anything heavier here or as a sediment in the centrifugation. And in which medium you are doing the centrifugation? Cesium chloride, right? So this experimental proof was given by the scientist Meselson and Stahl, and put his experiment with the help of which organism? Escherichia coli, right? Now, next after this, let's talk about the one more thing. If I am taking the concept of semi-conservative, this was even described by the scientist, that is a Taylor. The name of scientist was the Taylor and his co-workers. Right? So what they did in the experiment, here they use now radioisotope. Radioisotope of what thing they have used over there? Thymidine. And in which specimen? In the Vicia faba, which you commonly call it as a faba beans. Right? So even chromosomes at chromosomal level, they have described the semi-conservative process. That is done by the Taylor and his co-worker, but by using E. coli with the heavier isotope of nitrogen, that was done by Meselson and Stahl. Right? So this is about the experiment to show the replication is a semi-conservative process. Now let's talk about the machinery and the enzyme for replication. During the replication process, we need a set of enzymes. That means we don't want one single enzyme, we need many enzymes during that process. But most important will be the DNA dependent DNA polymerase. This is the name of enzyme. I will shortly write it at DDDP. Why DNA dependent? Because what is the sequence on the parent strand of the basis complementary to that will be on the replica. So what sequence of that basis will be there on replica, that depends upon the strand of DNA of parent type, right? So that's why you call it as DNA dependent. But here we are making new replica, which is of the deoxyribonucleotides. That's why you call it as DNA polymerase, right? And along with that, other even enzymes are included like helicases, which is unwinding the DNA and breaking the hydrogen bonds. Then is for the topoisomerases, which is relieving the stress of unwinding. That stress can break the DNA, but that stress will be overcome due to the presence of this topoisomerase, especially in case of the eukaryotes. And in prokaryotes, same thing is going to perform by the gyrases, right? And even in the last, we will discuss about the ligases, right? So all these enzymes, mainly what thing will be there? DNA dependent DNA polymerase, which actually make the monomers of the nucleotides to bind and give the polynucleotide chain, or you can say replica of the DNA. Fine. But during that process of the using this enzyme, where the main is this one, the process should be efficient and even it should be accurate. Efficiency means, or it should be efficient means, in less time, more bases are going to make its replica. For example, in your book, they have given the example of E. coli. They mention in E. coli, it is having 4.6 into 10 raised to power 6 base pairs. And they are replicating in 18 minutes. Just now I discussed with you. For replication of E. coli, it needs 18 minutes. 2 minutes is last for its cell division. Total 20 minutes for complete cell to divide into the 2 daughter cells, right? So I will take here 18 minutes. So as an average, we can say its efficiency approximately comes out to be 2000 base pairs per second. As per your NCERT book, you have to maintain this answer for this, right? So this is in less time, more base pairs they are replicating due to the presence of the enzymes. So it is an efficiency of this process. That is a replication process. Now coming to its accuracy. Accuracy means the mistake should not be there. Mistake should be avoided, or it cannot be, it should not be there, right? I must say here the mistake should not be there. Mistake should be avoided. If somehow, because it is doing the thing to the full DNA, here important thing, keep in your mind, the full DNA will replicate, right? And both the strand will show the replication. When you will discuss about transcription, only the part of DNA will transcribe, and out of two strands, only one strand will transcribe, right? So replication has to do much more. So mistakes somewhat can be there, but if it happens, then DNA, dependent DNA polymerase, one of its types, is going to proofread the full replica form on its parental strand. If any mistake happen, it will rectify that, right? So this even should be the accurate. Efficiency should be higher. And one more important thing you must remember here, this replication process is high energy consuming process. And due to that thing, the raw material, what is required there? Like for example, if I show you this replication fork, this is a parental strand and these are what thing? Replica or the daughter strand. To reach up to that stage, the high energy is required. So who has built this? The replica. Brick by brick, the nucleotides will come here. Which nucleotide will come here? Deoxyribonucleoside triphosphate. Are you getting that? Which phosphate is required here? Triphosphate. Usually nucleotides are having one phosphate. Like if I say here deoxyadenosine, actually it is monophosphate, but now two extra phosphates are coming with, that's why you call it now that thing as a deoxyadenosine triphosphate. Why word deoxy I have used here? Because you need it for the new DNA strand to form, not RNA. If it is RNA in transcription, then simply you can give the word adenosine triphosphate there, right? Here we are making the replica for DNA. That's why you have to use the word deoxy, except only at the thymine. There you can say thymidine triphosphate, because thymine is obviously in DNA. That's why. Even guanosine. In guanosine, you have to speak it deoxyguanosine triphosphate. For cytosine, what you have to say, or cytidine, I must say, deoxycytidine triphosphate. Right? So deoxy word must be there. And why triphosphate? Because two extra phosphate is there. And if you remember, in the biomolecules, even you have learned, and even in molecular basis of inheritance, this is a pentose sugar. On its 5' position, there is a phosphate. Here is a base. Right? Now, there two extra phosphate is going to be bound. Now, this is a triphosphate. Instead of one, now it is having two extra. Why wavy line? Because this is high energy bond. Once this bond is going to break, then high energy will be emitted, which help in the polymerization process of the replica, the newly formed daughter strand, opposite to the parental strand. So this thing is one, which is overcoming this demand. So can I say this deoxyribonucleoside triphosphate is working and the dual or playing its dual function? What is that? One will be, it is providing energy during polymerization. Second, it is even working as a substrate. While working as substrate, they are liberating energy, right? So it is having dual function, that is deoxyribonucleoside triphosphate. Ultimately, it will become monophosphate after the breaking of these bonds, right? But this phosphate is still there, so it will be a nucleotide. But monophosphate, deoxynucleoside monophosphate at that time, when these two phosphate are going to be leaving that after breaking of the bond. Fine. So can I say terminal bonds are going to break? Terminal means which is at the edge, not this one. It is not going to break. This one and this one will break. Fine. Okay. Now, what is next? If we are taking this process, the DNA is so big, so it cannot be happening from end to end. Suppose this is a long thread of DNA in hand of me, this long thread. I cannot start from one end and complete it to the next end. It is not happening like that. So how we overcome this thing? There are some specific site on the DNA from where the origin of replication must be done. That you call it as ORI. What ORI stands for? Origin of replication. Actually, suppose this is a double-stranded DNA which is having hydrogen bonds, right? This is a very big DNA. I am taking just a part. Suppose this is ORI point here. What will happen? The hydrogen bonds first of all will be broken with the help of helicases, and this will be open like that. Can I call this point now as ORI point? And you will get this Y-shaped structure on this side as well as this side. This you call it as this replication fork. But this replication fork actually formed two, but they have shown only one. So can I say this is ORI point? Even down you are having this Y shape, but only one they have shown in your NCERT book. And what they are asking, replication is bidirectional. And what students are thinking, it is drawn here, this direction, that's why it is bidirectional. No. From the ORI point, it is replicating on this side as well as on this side. Even if I am taking like that, downside, even upside. That's why it is a bidirectional process. Right? So, mostly it is a bidirectional. Rarely it can be the unidirectional. And ORI point in a prokaryote will be one, whereas in eukaryotes it can be many. It can touch to thousands even. Being the DNA is very big. End-to-end is not possible. So, specific point they are going to do that. And full DNA end-to-end is a very much high energy requiring thing. That's why the replication fork has to be formed. Right? Okay. Now, coming to the next point. After this, the opening happened due to the helicases, means at ORI, helicases unwind the DNA and break the hydrogen bond. Topoisomerase relieve the stress of the unwinding that will not allow them to break. Then SSBP means single strand binding protein. Much more other things you need not to worry. Simply you should know about the DNA dependent DNA polymerases, helicases, topoisomerases in eukaryote, gyrases in case of the prokaryotes, and at last ligases. At least this much. Although small little bit I am going out of NCERT with the word helicases, topoisomerases, gyrases. But you must remember that being it is given in the options or in the question, right? Okay. Now, once this is creating this fork, it is not like that directly red line means replica is going to be formed. To remember, all the convention you have to take from this open end. This is 3' of one parental, this is 5'. This is a fork end or the fork. If you are going like this, you are going away from the fork, but going towards the ORI point from where the opening happened, right? Now, remember one thing, replication is happening from 5' to 3'. New replica direction is from 5' to 3'. So, to give them the place to the new nucleotide, brick by brick they have to come. How they come to know that we have to come on this point? Because at this point, the primers are going to be added. At which prime end? 3' end of the parental strands. Once it is coming there, can I say they are having 5' on this side and 3' on this. This 3' is providing OH group. Why OH group is provided? Like for example, if I am giving you again this structure, this is one nucleotide with its base and the phosphate. This is next one. This is even having its phosphate, right? Suppose this is the thing. Nucleotide to nucleotide. If polymerization is required, you remember at 3' end there is OH. At here with the water loss, there is a formation of phosphoester bond. This is one phosphoester, this is second phosphoester, this is diphosphoddiester bond. I repeat this point once again. One nucleotide, second nucleotide, how they will bind with each other at 3' end because there is OH group here. This OH group will provide a place to make the phosphoester bond between two adjoining nucleotides. This nucleotide already having phosphoester bond. A new phosphoester bond have been created. That's why together you call it as phosphodiester bond. which will help in the polymerization of one nucleotide with the next. So primers, what is the main role of that? They are providing the OH group over there. Is that clear? So this is very important thing that you should know what is the role of primer. And do remember, this primer is RNA primer. Right? This is the RNA primer. These are RNA primers. But once the things are coming one after the next, like if I am taking here one after the next, if the next nucleotide come, next will come, so on. How they will polymerize with each other? No doubt with the enzyme DNA dependent DNA polymerase, right? But once the polymerization started, the extra 2-phosphate from every nucleotide will be going to be released by the breaking of bond, and that energy help to create the phosphoester bond and help in the polymerization process. And obviously there is a creation of hydrogen bonds even between the replica and the parent. And you see the replication is happening from 5' to 3'. Like here even they have given 5' to 3'. The replication happens. But problem happens here where the parental polarity is 5' at open end. Here no doubt it should be 5' to 3'. But here it will be discontinuous formation of the replica. And every time you need the new primer here. Like if they have shown 3 fragments, which even you call it as Okazaki fragments, right? There that very number of the primers are required. And after that what will happen? These primers are going to be removed. And that gap is going to be filled again by polymerase. They are one another type. But Okazaki fragments, the Okazaki fragments will be now linked by which enzyme? Ligases. Ligases here will be binding Okazaki fragments with each other. So ligases you required only in this discontinuous synthesis. It is not required in continuous synthesis. So this side will be the leading strand, this side will be the lagging strand. Right? But polarity will be 5' to 3'. Here even 5' to 3'. Are you getting that? So you must remember all the enzymes and how the polymerization happen, what is the role of a substrate and which direction replication is happening, and DNA full is replicating or a part. Obviously it is a full DNA. Will both the strand replicate? Yes, both the strand will replicate. One in discontinuous manner, other will be in a continuous manner. Because polarity must be 5' to 3'. So in that thing, this type of the complexity happen. It will be broken in the form of Okazaki. Fine. And you remember in cell cycle cell division, where this replication happen in eukaryotes, in S phase, during cell cycle and cell division. And next, if after this interphase where S phase happened, replication happened, but after that, no karyokinesis, no cytokinesis, only replication keep on happening. It will create the ploidy, right, which is an anomaly. Do remember that fact. More detail of cell cycle, cell division. When you will learn, you revise, I hope so, you will come across that after the interphase, karyokinesis and the cytokinesis is must. Interphase must be followed by the cell division. Fine. Okay, now let's talk about some of the questions, PYQs, which have been appeared in few I am taking here. Now, here I am taking the first question, complete the flow chart on central dogma. Just now I discussed with you what A stands here for the replication. So, can I say we are now in doubt with 2 and 4 because A here is given transduction, here translation. It is not like that. So, we are left with 2 and 4. Let's take it on the B now. It is the DNA forming mRNA. Definitely it is a transcription. Let's check the C now. This is mRNA forming the what thing? Obviously the proteins here. So we have figured out 2 things. C is for the translation, D is for the protein. So who is mentioning that thing? D. So our final answer here will be D. Let's check the next. The DNA dependent DNA polymerases catalyzes in which direction? Only 5' to 3' direction. Just now I said, even it gives the complexity. That's why one strand will be showing the continuous formation, other will form the discontinuous. That's why we call replication even as semi-discontinuous or semi-continuous process as well, right? Now coming to the next question, what is given here? E. coli has only 4.6 into 10 raised to the power 6 base pairs and completes the process of replication within 18 minutes. Then the average rate of polymerization is approximately, this is a direct line from your NCERT, even we discussed in our lecture that it should be 2000 base pair per second, right? So here for this question answer will be first option, right? So likewise, please do revise your topics and practice some multiple choice question. Only then you can verify that where you are perfect or where you have to practice more, right? So this is all for today. I hope so you will revise that and we will meet next time with the process of the transcription. Thanks a lot.